Hello students, today we will discuss the structure of anther. I have already discussed about the structure of stamen, which contains two parts, anther, filament. So when we take the section of the anther, we will find the structure like this. Having four sac-like structure. So basically, if we divide the anther from this line, this is a lobe and this is a lobe, so we can tell bilobed anther, and each lobe contains one thecus. This is a thecus. So these two can be written as dithecus. So each lobe is dithecus and it is bilobed anther. So these thecus or the cavities are called as microsporangium. Plural form microsporangia. Plural form. So basically we can think how these microsporangium are produced, as well as this dotted structure which are called as microspore or pollen. So in young or tender anther, we will find the structure like this, where there is no differentiation of any thecus or the microsporangium. So you will find only the mass of parenchymatous tissue. Mass of parenchymatous tissue. Mass of parenchymatous tissue. Then in the next step, gradually what happens? There is differentiation, and just below the epidermis, what happens? There is specialized or differentiated cells which will in future gives rise to the microsporangium, that is called archaeosporial initial cell, and is identified by the dense cytoplasm as well as nucleus, which is differentiated from other cells. So next it will gradually divide into two cells. The outer is called primary parietal cell, and second one, inner one, is called as primary sporogenous cell. The primary parietal cell produces wall layer, primary sporogenous cells produces pollen. So this next structure, the primary parietal cell divides in all directions and produces the wall layers. Outer epidermis, then endothecium, then middle layer, and innermost is the tapetum. And inside the cavities or the sac, before formation, it is filled with large number of cells called sporogenous cell or pollen mother cell. PMC, pollen mother cell. The formation of pollen is called microsporogenesis. So microsporogenesis, in this process, the cells which are present inside the innermost is called PMC, pollen mother cell. This pollen mother cell is 2N or diploid in respect to chromosomes. So this pollen mother cell, it will undergo meiosis. So this division we can write meiosis 1, and it will divide into 2 nuclei after karyokinesis, and later on it will divide into 4 nuclei after meiosis 2. So here the nucleus contains haploid number of chromosomes, and after that what happens, the cytokinesis takes place. And they produce the pollen tetrad. Having haploid pollen. This is called pollen tetrad. And this pollen tetrad segregates to form the pollens. And having what? The outer layer exine made up of sporopollenin, which is resistant to decomposition, and inner is called intine. intine made up of pectocellulose. So at the time of dehiscence, normally the pollens are in two cell stage. One is generative cell and another is vegetative cell. In future, this generative cell gives rise to two male gametes, and vegetative cells behaves as the tube cell. So this process is called microsporogenesis. Then the process called development of male gametophyte. So this male gametophyte development occurs after the pollination process, and here what happens, normally the pollen falls by the process of pollination. We will discuss detail in pollination later on. So the pollen tube will extend through the germ pore where exine is absent. So inside the style, what happens, normally the generative cell divides into two male gametes, and tube nucleus or generative cells behaves as the tube nucleus. Okay.